Hello. Hello, and welcome back to our summer Lunch and Learn lecture series at the Harvard Center for Biological Imaging. It's been quite some time since we've done one of these, so it's exciting to see all of you here and to get back into the swing of things. So usually when we do these, we start with something a little more introductory and easy at the beginning and then build up. But we're doing that a little bit different this year. We're going to dive right into the deep end with something that's pretty complex. And so today what I want to talk to you about is super resolution imaging, the field of fluorescence microscopy that's become pretty popular in the last few years. And in that field, I want to talk about one technique in particular, a technique called STED. So before we get right into that, though, because like I said, this is the first lecture of the year, there's definitely some background that we need to talk about first. And so obviously, we're going to be talking about fluorescence microscopy. So we need to know a little bit about fluorescence before we can dive right into STED and see the mechanisms behind it and how it really works. So if you're not familiar with the real hardcore physics, not even the hardcore physics, but the basic physics behind fluorescence, I definitely recommend that you read this review by Jeff Lichtman and Jose Concheo. It came out many years ago, but it's a really good summary of how this process works. So I think we're all familiar with seeing these absorption spectra and emission spectra for particular dyes. This looks like something in the green range, like maybe an Alexa 488 or something like that. And what's always interesting about these is that there isn't, although we call it, say, Alexa 488, it doesn't absorb just at 488 nanometers. That's where its peak is. But there's all these other wavelengths that that molecule can also absorb light at. And you can see even down here, and when you get into the UV range, there's another peak that's coming up here as well. And likewise with emission, these dyes don't give off at a single wavelength. They give out over a range of wavelengths, and this can be quite broad. Obviously, this tail goes on for quite some amount of the visible spectrum, and these actually give off over well over 100 nanometers at different wavelengths of light. So why is that? Well, to really understand this, we need to look at something called a Jablonski diagram. And a Jablonski diagram describes the energy levels that a particular molecule can occupy depending on the amount of energy it absorbs. So if we think about fluorophores, they're these very complex molecules. They have these intricate ring structures, they're large molecules, there's a lot of electrons in very complicated configurations around these molecules. And what that means is that these electrons, especially the ones in the outer shell, are able to absorb energy, and that can be absorbed in the form of a photon. And so these diagrams, Jablonski diagrams, try to describe the different energy levels that one of these molecules can occupy. So the S0 level right down here, this is the ground state. This is the resting state of that molecule when it hasn't absorbed any light. And then, because this is quantum physics, there's only certain levels of energy that each one of these dyes can occupy. So you can see we have this group of energy levels here called the S1 energy level, and then we even have one way up here, the S2. And these energy levels obviously increase in energy the higher you go up here. So the S2 level has more energy than S1, and that's more energy than the ground state, obviously. But what's interesting is light, obviously, or photons, have different amounts of energy based on their frequency, their vibrational frequency. So you can have a UV photon, so something here down here in the UV range, close to, say, 325 nanometers, that has a lot of energy. So if this fluorophore absorbs it, it goes up way up to one of these really, really high energy levels. Right? However, you can see it can absorb basically anything along this absorption spectra. So we could have something closer to what we'd call a green photon over here. And you can see that doesn't have as much energy as the UV range. So it only brings it up into this S1 range. Okay? And obviously we can go up to a whole bunch of these different levels. Whenever one of these fluorophores gets excited and gets into these excited states here, they then give off energy, and they give off by vibrating, by rotating, and they lose some of that energy until they fall back down to the lowest energy level in the S1 state here. Once they get to this energy level, and only from this energy level, can they transition back into the ground state by giving off another photon. And so again, we have an emission spectrum, which means there's multiple different wavelengths that they could give off. So for example, this one could drop back down here by giving off a green photon, very similar to this green here that could have been absorbed. And you can see if it gives off this one, it drops all the way back down to that lowest energy state, the ground state. But just like in our process of going up and our process of going down, we don't have to go all the way to the lowest energy level of S0. We can go to a higher energy state in S0. So for example, we could take this, we could give off this orange colored photon and drop to the near the highest energy level in S0. And then again, once here, you could have some rotation and vibration and eventually get down to that lowest energy state. So hopefully this is a better pictorial representation of why we have these broad spectrum and we just don't have a single peak at an absorption wavelength and a single peak at our emission wavelength. But both the absorption and the emission are a spectrum. So there's a wide range of wavelengths that can be used to excite a fluorophore, and likewise it can give off a bunch of different wavelengths as it falls back down to its ground state. Okay, so that's the basics on fluorescence, and we'll come back to one of those Jablonski diagrams, or to that same one a little bit later. So the other thing we need to understand real quickly is just how a fluorescent microscope works. So fluorescence microscopes, in their most basic form, is what we call a wide field fluorescence microscope. And there's several super resolution techniques that are based on wide field fluorescence microscopes. So structured illumination is based on a wide field scope. Single molecule localization experiments can be done with wide field scopes. And basically all you need to know here is that we have some sort of light source. In this case, we've got a white light source that comes in here, and it gets sent into our filter cube. And on one side of that filter cube, we have an excitation filter that filters out, in this case, just the green light. So this is going to be a red fluorescent dye. So it filters out just the green light from that white light that's coming in, and let that green light come in and bounce off this dichroic mirror here, which is a very special mirror that will reflect green light but let other colors of light pass through. So that green light gets reflected off that dichroic mirror down onto our sample. We create some fluorescence. Fluorescence goes back into the objective. This is now red fluorescence, and it travels up. And again, I said this mirror only reflects green light, so that red light is going to pass through, and it goes up to the eyepiece or the camera. There's an additional emission filter here as well, just to clean up some of that light, just in case there was some white light that got through here, or some of whatever this green light, any of this green light that reflects off our sample and comes up, and if some of it gets through that dichroic mirror, we just do an additional cleanup there. Right? The other important thing about this, and it's kind of hard to tell, but if you look at the green light that's coming down here, you can see it's being focused. So it's going to get focused to the back focal plane of the objective here. And then that means it's going to come out as a collimated beam on this side. So this is why we call this a wide field microscope, because the light comes out in a wide field. The excitation light comes out, floods the entire field of view underneath that objective. All right. So wide field microscopes have some advantages and disadvantages. The biggest advantage is obviously that this is really fast. So here's our objective lens. Here's that collimated light coming out of it. And you can see our entire field of view here is bathed with this blue cyan excitation light. Unfortunately, though, not only does it bathe it in the lateral dimension in X and Y, but it also bathes it in the axial dimension. And so that means all of these fluorophores that we have here from the top to the bottom of our sample are all being excited. And even though we only want to image these three that are on our focal plane, that's what this white line here is, the focal plane, we've got all these other ones that are being turned on as well. And so that means if we have the thicker our sample is under a wide field microscope, the blurrier our image is going to look, because we're going to get more and more of this out of focus light also contributing background haze to our image. So this is a major limitation of wide field microscopes, is that they only work with nice thin samples, and the thicker your sample gets, the more blurry it's going to get. And another restriction, and what we're really going to focus on today, is that all light microscopes, microscopes that work with excitation light and emission in that visible wave range, wavelengths of the visible wavelength, are all limited in resolution. Okay. So I said there's some types of super resolution that are based on wide field, but the technique we're going to talk about today is STED, and that's based on a confocal microscope design. So a confocal microscope has three key differences between from a wide field microscope. So the first one is our excitation light here, instead of getting focused at the back focal plane of our objective, it actually comes in as a collimated beam into the objective. And what that means is that when it comes out the other side, it's going to get focused to a point. So in a confocal microscope, in this case, this is a specific type called a point scanning confocal microscope. We're focusing that light to a single point. We're really only imaging our sample one pixel at a time. Right? So that's the first big difference. Right? So that's all great. This light is going to get focused. If we've got a little fluorescent molecule here on our sample plane right here, it's going to get excited. But that light comes in through our sample on this side, and then once it gets to the focal plane, it's going to start diverging on the other side. And we still have this issue where if we have fluorophores above or below that point that we're imaging, they're still going to get excited. So we are still going to have some background out of focus light contributing to our sample and degrading our image. So we need a way to get rid of that. So this is hard to see, but this green dot here, these green lines are just the ray trace from that spot going back through our microscope. So the light from this point is diverging as it goes into our objective. Our objective turns it into parallel light, sends this parallel beam of light through infinity space in our microscope. It can pass through the dichroic mirror, because it's a different wavelength than our excitation light. Then it gets to the tube lens in our microscope, where it gets focused again. It gets focused to a point. Okay? If you have a fluorophore that's a little bit lower, and although I've colored this one magenta, it's meant to be the same thing. So it's meant to be a green fluorophore as well. I just wanted you to be able to see it separately from this green one. And so if we do a ray trace from there, same idea. We have diverging light that gets captured by our objective. We get parallel light going through to our tube lens. But then when our tube lens focuses this light, you can see it gets focused a little closer to the tube lens, further in front of where the light from this focal plane comes. And so here's the big second difference in a confocal microscope. So a confocal microscope, you've probably heard about the pinhole. Confocal microscopes have a pinhole. So that pinhole gets placed right here, where the light from any fluorophore that's in the sample plane gets focused over here. So this is what's called a confocal plane. So the light that's in focus here is also in focus here. But you can see that light from the magenta spot now gets focused over here, and then it starts diverging again. And what that means is that most of that light gets blocked by the outside of our pinhole here. Okay, so it just hits the surface and none of it goes through to our detector. And of course, the detector is the third difference. So with a wide field microscope, because you're bathing that entire field of view in light, you can just send that to a camera or to your eyeball and take a picture or see it immediately. In a point scanning confocal, all we're looking for is whether there's light or not light and what the intensity is at that one spot. So we don't need a camera with a million pixels because we're only looking at one pixel. So we use some sort of point detector, and what this usually is something called a photomultiplier tube. I won't get into that today. We can save detectors for another day. But one more thing that's important is, so right now I've showed you how to image this one green fluorescent spot that's right here. What if we have something that's out away from the center, somewhere else on our focal plane? How do we image those? Well, the way we do that is inside our microscope, there's a couple mirrors, and those mirrors can move back and forth. And if we tilt the angle that this light is coming into our objective, that changes where the light gets focused on our focal plane. So you can imagine if we're tilting this way, we're either tilting to the left or to the right here, we're going to scan up and down this line. Whereas if we tilted in or out of the screen, then we would likewise scan out or in of our screen over here. So by having two mirrors in this light path, I haven't shown them here, something like this, two mirrors that we can tilt our beam in both directions, we can now scan it back and forth across our sample. This is something called raster scanning. Okay. And if you do that, you get this standard image acquisition that we're all used to in a confocal microscope, where we slowly scan back and forth, and build up our image over time. Okay. So that's our confocal microscope, how our confocal works. Again, there's some disadvantages and advantages. The big advantage here is we have improved contrast and we're able to now acquire beautiful 3D images that we just can't really do with a wide field microscope on thicker samples. Disadvantage is going to be slow. We just can't click that one image on a camera anymore. We have to wait to scan back, raster scan back and forth across our sample 1 pixel at a time, so it's going to be slow. And again, we still have this limited amount of resolution. All right, so today this is what I'm going to talk about for the rest of the time. We're going to talk about breaking this resolution limit and why it exists. So the diffraction limit has existed for a long time. So Ernst Abbe, who was one of the first employees at Carl Zeiss Microscopy, way back in the mid-1800s, came up with this equation right here. And this equation defines what the resolution limit of a light microscope is. And this is due to the wave nature of light and trying to pass that light through small apertures causes it to diffract. And so what he said was that the distance between two objects, the smallest distance that two objects can be and still be resolved by your microscope, relates to the wavelength of light that you're using to image and the numerical aperture of the objective. That's what this n sine alpha is. So today we usually use this equation, a little more modern version of this, but we basically say that resolvable distance of two objects is equal to 0.61 times the wavelength of light that you're using over the numerical aperture of the objective. And this is in the lateral dimension in x and y. I'm not going to talk about axial dimension or z today. So, like I said, Ernst Abbe first defined this back in the mid to late 1800s. And basically what this says is that when we have a single fluorescent molecule inside our microscope and we excite it and it gives off light, and we're looking through the eyepiece at it, or we're looking on the camera at it, it doesn't look like a point source anymore. By the time it gets to the camera, it's diffracted, which means it's spread out. So it becomes this blurry spot with all of these concentric rings around it. And as long as these are far enough apart from each other, it's no problem. So like right here where it says resolved, I can see that there's 3 different spots there. But at some point, they start to get really close together, and it's harder to tell whether that's 1 or 2. And at some point, they get so close together that there's no way that you can actually say with certainty that this was 2 objects over here. It looks just like 1. So I'll just give you maybe one quick example of this. We'll just do the math on this equation. So here's our equation, just another variation of it, lambda over 2na. So if we plug in 525 nanometers as our wavelength of light, so we're looking at green light, and let's say we're using this objective here. This is one of the best objectives we have in the facility. This has a numerical aperture of 1.4. Notice that magnification doesn't mean anything here. It doesn't matter at all. So whenever you say what the magnification of your objective is, that means absolutely nothing to me as far as what you can resolve. What's important is this numerical aperture, the little number down here. So we plug that in at the bottom, and you can see here that we can resolve structures down to about 200 nanometers. Right? So again, that's cool, but what does 200 nanometers actually mean? So let's take a look at this electron microscopy image. So this is electron microscopy image. Here's the plasma membrane of the cell. I think this is the edge of the nucleus over here. And here are 2 endosomes. These 2 round structures here are endosomes. And I think you can see, hopefully you can see in this sort of lower left side of the lower endosome here, there's this black spot. And this is a sample that had immuno-EM performed on it. So this is a 10 nanometer gold bead that's been attached to a protein of interest on the surface of this endosome. Conveniently, 10 nanometers is about the size of a GFP protein. It's just a little bit bigger. But let's say that instead of a gold bead here, and there was another one over here too. Let's say we actually had 2 molecules of green fluorescent protein. Right? So that's what they actually occupy in real space. But if this microscope was a fluorescence microscope instead of an electron microscope, here's what those 2 GFP molecules would look like on our camera. Just 2 big blurry spots. There's no way that you could actually tell that these were 2 different molecules. And you certainly couldn't tell that there were 2 endosomes here. Even if there was GFP molecules all the way around the edges of them, you just see 1 big spot. All right? So this is the problem. Obviously, biology happens below this diffraction limit, and there's some certainly many processes and reasons why you would want to be able to resolve and see at this level. So we need to somehow do better than this. So I told you Ernst Abbe came up with this equation way back in the mid to late 1800s, and that's the way it was for many, many years. But surprisingly, just a couple months apart in 1994, 2 different people, Stefan Hell and Eric Betzig, came up with 2 proposals for ways to get around this diffraction limit. So Stefan's method was STED, which is what I'm going to talk to you about today. And then Eric's method was PALM or STORM, or a family that we now call single molecule localization. So in '94, they came out, these papers were both just theory. There was no experimental proof here, but as we all know, a few years later, they did do experimental proof of it. These technologies have now been commercialized, and they eventually won the Nobel Prize for it back in 2014. So let's dive into STED, one of these super resolution techniques, and see how this actually works. All right, so here we are. We've got a whole bunch of fluorescent dye molecules that are all sitting close together. So these are within 200 nanometers of each other. Now, if we put these in a point scanning confocal microscope, we turn on our laser to scan it back and forth across our sample. When it lands on these, obviously they're all within that excitation spot because that excitation spot is diffraction limited. This is as small as we can make it, is about 200 nanometers, even when we're using that really expensive, awesome objective. This is the best we can do. And of course, all of those molecules all get lit up. Everything turns on. But wouldn't it be awesome if somehow we could take some sort of magical donut and put that donut over top of our excitation laser, and only fluorophores that were on the inside of that donut hole were able to be seen. All the ones on the outside just disappeared. And then if we were able to move this donut with our excitation spot back and forth across our sample, we'd have much higher resolution because we'd only be collecting from the center of this donut instead of the entire donut. And this is basically what STED does. So now you can see we have a much higher focus on that, that laser beam. And effectively what we're doing is shrinking the size of that excitation laser spot that we're moving across our sample. Okay, so you can see now it looks like it's only this big, it's only the whole of that donut. Okay, so that sounded like fantasy, right? Well, it turns out we can actually do something quite similar to that. So if we go back, here's that diagram that I showed you earlier of a confocal microscope. Everything's exactly the same here, except I removed that magenta spot because we don't care about that anymore. So if we wanted to turn this into a STED microscope, there isn't really much that we have to do. All we have to do is add another laser beam. So we've added now this orange laser that's come in. We have to add another dichroic mirror to shine that, to bounce that orange laser down onto our sample. And then we're going to put in an optical component here, some sort of polarizer. It could be a spatial light modulator. And this is going to shape this orange laser beam into a donut. Okay? So one of the first and initially most commonly used optical elements for doing that is something like this. This is often called a vortex wave plate. And so this is a piece of glass, and if you go around the circumference of the circle, the thickness of the glass in this wave plate is always changing. So what you can see is sort of right here, if we want to call this our starting point, or say 0 degrees right here, it's really thick. And as we go around, it gets thinner and thinner and thinner. At this is 180 degrees. It's half as thick as it was over here. And as we keep going, sorry, that's not true. It's less thick than it was over here. And when we get to the end here, you can see it's a little bit thinner as well. And so what that means is that light that goes through one side of this vortex wave plate. So let's say we had light going through right here, and light going an equal distance and direction from the optical axis over on this side. Those two beams of light would be exactly out of phase with one another. So what this does is it just creates a phase ramp across your beam of light that's going through this phase plate. And it creates a phase ramp where light on opposite sides of that cylinder of light are exactly out of phase with one another. And what that means is when you focus those down to a point, they interfere with each other at the focal point. So you get total destructive interference at the center, and all of that light energy is pushed to the outside of your focal spot. Okay? So instead of focusing all of that light to a spot and jamming all of the light that's coming from our laser through our objective into one little 200 nanometer spot, now what we've done is we've created a laser donut with a hole in the middle of it. All right? And if we take these and put them on top of each other, Then what we have is excitation in the middle, and then we've got this weird laser donut around the outside. And this laser donut, for right now, I'm going to say it turns your fluorophore off. That's not entirely true. I'll show you what it actually does in a second. But for right now, let's just think we have an excitation spot. So we have one laser that's giving us excitation light. We have another laser that's giving us this STED donut. This one turns our fluorophores on. This one turns it off. If we put those right on top of each other, only the fluorophores that are in the middle are actually going to turn on. And so as long as this hole in the middle here is smaller than the diameter of this donut hole is smaller than the diameter of this excitation spot, we're increasing the resolution of our microscope. And so you can see here we've gone from, say, our 200 nanometer excitation spot to something, an effective excitation spot of around 70 nanometers. Okay? And the way you control the size of this donut hole is by controlling the power of this laser. So as you increase the power of this orange laser, that hole in the middle of the donut gets smaller and smaller. Okay? Cool. And that means your effective fluorescent spot gets smaller and smaller and your resolution gets better and better. Yeah? Okay, so I said this orange donut laser is turning fluorophores off. That's not true at all. So to explain what this one's actually doing, we need to go back to our Jablonski diagram. So let's say we want to perform STED microscopy on this green dye here. Now, I'll show you later, we usually don't use green dyes, but that's fine. So what we're going to need is we're going to need two lasers. So we're going to need one laser that's going to do our excitation. So let's choose something close to the excitation peak of this dye. So we're going to use this green laser here, or the cyan-y greeny laser. And you can see that's going to excite our dye up into the excited state here. Perfect. Now we are going to need our STED laser. And so for our STED laser, we're going to choose something out here in the orange, somewhere out on the tail of this emission spectrum. And you can see that's out in this space. And so I told you before that this was turning the dye off, and that's not true. What it's actually doing is it's forcing any excited molecules to fall back to their ground state by giving off a photon of exactly this wavelength of light. So usually once this fluorophore gets in the excited state, it has all these different options, all these different arrows that it can fall back down and give off everything from a green to an orange photon. But through a process called stimulated emission, which is exactly how a laser pointer works, if you are in this excited state and another photon comes in very close proximity to that molecule, it can force it to fall back down to the ground state by giving off a photon of exactly the same wavelength. So if we bathe this excited sample in this orange light, it's going to force that excited molecule to come back down into the ground state via this orange arrow. It won't allow it to come down to the green one or one of these yellow ones. It has to come down in the orange. So in the center where we have that excitation laser spot, we just have the standard thing. We're exciting our fluorophore up here. It can take any one of these arrows back down to the ground state. But in that area where the STED donut beam is, that only allows the light to come back down via this orange one. So we can only come down this orange path. We can only give off an orange photon. Any fluorescent molecules in this orange donut space cannot come down the green or the yellow arrows. And so now if we put an emission filter in our microscope in front of our detector that only detects these green and yellow arrows and does not detect this orange arrow, that means we're only going to collect the light from the middle of this spot. and we're not going to see anything, any of the light that's coming back down on that orange arrow. So now we only, although all these molecules are getting excited, all of them are giving off fluorescence, only the ones that are in the circle in the middle are actually being detected by our detector. Okay, so that's how we effectively shrink that excitation spot. Cool. So one more thing that I should say about this, um, you can get, uh, so obviously I said before that the, um, the size of that donut hole gets smaller the more powerful your laser is. So obviously we want to use a high-powered laser for our STED beam, and that can create some problems. You can have some phototoxicity if you have a live sample. You can have some bleaching even if you have a fixed sample. Um, so you want to be really conscious of your photon budget and how many photons you're pumping into your sample. And because of this, pulsed lasers are absolutely essential for getting the best STED results. And so if we think about this in time, this initial absorption of a photon takes about 1 femtosecond. It's really, really quick. This vibration and rotational relaxation takes about 1 picosecond, slower than a femtosecond, still very fast. But then molecules tend to hang out in this lower energy state of S1 here for a few nanoseconds. Yeah? And so obviously that's a little bit longer. And especially when it comes to pulse lengths of most pulsed lasers, they're not nanosecond pulses. So usually for a STED microscope, you not only need a pulse laser, but you need a pulse laser with a really long pulse length. And the other thing that you want to do is your excitation laser is also pulsed, and you want to get these two, the timing right. So you want to make sure you get everything pumped up into this excited state before that STED laser beam comes in and forces the sample down this orange arrow. So this is probably a little hard to see, but basically in this diagram, green is your excitation light, your excitation pulse, and red is your STED pulse, and then the yellow is showing you how much fluorescence you're getting. So you can see here that there's a pretty big gap in between when that green excitation pulse comes and when that STED pulse comes. You can also see the STED pulse is a lot wider than that excitation pulse. But because this is too late, you're still getting all kinds of fluorescence. So that yellow fluorescence emission intensity is still really high, okay? But if we move the STED beam a little bit closer to that excitation pulse, so we get the excitation pulse and then our STED beam is peaking in intensity right after it, you can see now we've knocked out all of that fluorescence with the STED effect. You know? So this is really important. So it's important that your excitation pulse matches the absorption and vibrational relaxation timing. Your STED pulse should match close to the fluorescence lifetime. And your STED pulse needs to immediately follow that excitation pulse. The other thing that's really good to know about when you're using these pulse lasers, you can see they're off more than they're on. So this x-axis here is time. That STED laser is only on where these red lines are. So you can see that it's actually off more than it's on. And that's going to have a great effect in reducing your bleaching and phototoxicity in your cells. Okay, cool. Okay, so now we put it all together. So basically we now have our excitation beam. In this diagram, it's the green one in the middle. We've got our STED beam, the orange one around the outside. And all we're going to do is raster scan this back and forth across our sample, just like we do with a confocal. And now we have a much smaller effective excitation spot. We're only collecting light from that green area, and therefore we have a much higher resolution. And that's how STED works. That's really all you need to know. Um, so just a couple things about our facility. So at the Harvard Center for Biological Imaging here, we have an Iberia Steadicon system on a Zeiss Axio observer stand. Um, so this system can do standard confocal imaging. We've got 4 excitation lasers that we can use, 405, 488, 561, 640. And we have 1 STED laser line on here. It's a 775 nanometer line. We get resolutions down around 30 nanometers. All right. So although we have all these excitation lasers, we're only doing standard confocal imaging in the blue and the green channel. But I'll show you how we can do 2 color STED imaging, even though we only have 1 STED laser here. And the secret to that is your dye selection. All right, so your dye selection is absolutely essential to making this work. As far as objectives, we've got a 100x 1.46 Na oil objective, and we also have a 63x 1.15 water objective. Both of these can be used. The water objective is really nice. It has a very long working distance, so it's good if you want to combine this with, say, expansion microscopy or live cell imaging. Okay, so this is how our particular system works. So here's our STED laser here out at 775. And then we've got 2 excitation lines, 561 and 640. And here's our detection ranges. So we've got 1 detector that's centered around 600 nanometers, and we have another detector that's centered around 660. Right? So Iberia, the company that makes the microscope, they also make some dyes. So these are the dyes that we certainly recommend. So there's one called Star Orange and one called Star Red. And you can see that both of these peak. So Star Orange peaks within detector 1 and Star Red peaks within detector 2. And what you can also see is that the tails of both of these dyes go all the way out to the 775. Okay, so this is what our STED laser is going to drive the emission of any excited Star Orange or Star Red dyes. Is going to drive them to send off that emitted light at 775, well outside of the detector range of either of these detectors. Okay. These are not the only dyes that this works with. We know of a few others that have been tested, so you can definitely come to us and talk to us or take a look throughout the literature. Here's an example. This was actually done with Alexa 594 as well as a previous Iberia dye called Star 635. So this is looking at axons and neuronal cell cultures. You can see we've got spectrin and alpha-abducin labeled, and on the confocal, it just looks like pretty much white. You'd say these are very well co-localized and maybe interacting proteins along this actin. But once we view those with the super resolution and view it with STED, you can actually see they're very unique domains that alternate back and forth, green, magenta, green, magenta. And you see these stripes that are formed along the axon. So this is one example of what you can look at. I like to think of STED microscopy as opening up. So we have confocal opened up subcellular imaging for us. We're able to start to look at ER and Golgi and endosomes and things like that. I like to think of STED and the other super resolution techniques in light microscopy as opening up suborganellular imaging space. So now we can start looking inside the Golgi, inside the mitochondria. We can look at nuclear pore complexes. We can look inside focal adhesions, various cytoskeletal components. So we can really start to see these inside these organelles that are certainly far beyond the resolution limits of a standard confocal or wide field microscope. Great. So this is another great review. This is also out of Stefan Hell's lab by Stefan Stahl. And I'll just summarize by stealing the summary figure from that paper. And so these are point spread functions of various microscope techniques. So Here's our standard confocal point spread function. So this is showing axially, so this is the z dimension up and down here. And so what we talked about today was STED. So you can see we didn't talk about improving anything in the axial dimension, but you can see now how much skinnier this point spread function is and how much better our resolution is in the lateral dimension in x and y now. Okay? So really kind of exciting stuff. And obviously, if you have any questions or want to start delving into some of the other techniques that are on the screen, we're able to accommodate some of those as well. Okay. So thank you very much for your attention. Thanks for coming back. It's exciting to be doing these again. We'll get this up on our YouTube channel as well, and we'll hopefully have a few more videos popping up there in the near future. Okay, so we'll see you back here in a couple weeks.